Okay, so let's get started. So welcome everyone, and thank you very much for joining us for this second Journal of Physiology virtual journal club ever. As we gather for this meeting, physically dispersed and virtually constructed, let us take a moment to reflect on the meaning of place, and in doing so, recognize the various traditional lands on which we do our business today. We acknowledge the elders, past, present, and emerging, of all the land we work and live on, and the ancestral spirits with gratitude and respect. So to start with, I'd like to thank the organizers for giving us the opportunity to facilitate this webinar today. In this time of social distancing, it's very much appreciated that we're given a chance to discuss research, share knowledge, and network with colleagues from all over the world. So thank you very much to the Journal of Physiology for this very welcome initiative, and also a special thanks to Rosie and Imran for all their help organizing this event. So just a little bit of housekeeping to start with. So for this webinar, there will be 3 presenters who I will introduce shortly, and we'll be presenting for about 30 to 40 minutes. And at the end, the audience will be offered to ask questions. So you're very welcome to submit your questions throughout the session using the Q&A button that should be at the bottom of your screen. And if you want to, you have the option to ask questions anonymously. There's a tick box for that. But if not, it would be great if you could also name your institution when you ask your questions so that we can read it out loud to exactly as you would do as a conference if you stand up to ask a question at the end of a presentation. So note that using this chat function, you can also, this Q&A function, sorry, you can also upvote questions, meaning that if you see one that you'd like to be answered in priority, you can, I think, give it a thumbs up. And this also stops the same question from being asking more than once. This session will be recorded and it will be emailed to you within a week. So you're more than welcome to share it with colleagues who couldn't attend today. And finally, I'd like to draw your attention on the fact that there will be a networking session after this meeting. The link will be separate to the current one and it will be posted in the chat box at the end of the meeting. Please consider joining the networking session if you can, because again, it's a great opportunity to keep the discussion going and to network with colleagues from all over the world. Think about it as if it was, the drinks part of the conference. There's no way you would not go to drinks, so come to the networking session. The paper we will discuss today is entitled The Effect of Sleep Restriction with or without High Intensity Interval Exercise on myofibrillar protein synthesis in healthy young men. So this study was led primarily by a group located here in Melbourne, in Australia. And the first author is Dr. Nick Steiner, who is one of our panelists today and who I will introduce shortly. So let's get started. So I am Dr. Severine Lamon. I'm a senior research fellow at Deakin University School of Exercise and Nutrition Sciences, also in Melbourne, Australia. And I will be chairing this presentation today. So with me, I've got 2 panelists today. So Dr. Nick Steiner, as I mentioned, is the first author of the study that we're going to discuss today. And this study was part of his PhD that he completed at Victoria University under supervision of Professor David Bishop. And he's since then graduated from his PhD and moved on to his starting his postdoctoral training at the Baker Heart and Diabetes Institute, also in Melbourne. And we also have Olivia Knowles. So Liv is a PhD student at Deakin University, supervised by me and others. And Liv is here tonight because her PhD investigates a very similar but slightly different question as Nick's question, looking at the impact of sustained sleep restriction on various muscle health outcomes. And Liv also authored first and sole author paper, a journal club paper, discussing and commenting the results from Nick's paper, which was also published in the Journal of Physiology a couple of months ago. So I think we're nearly ready to start. Please do not hesitate to share your thoughts about this virtual journal club meeting using the hashtag #JPJournalClub on Twitter if you'd like to. So a little bit of geography to start with. Nick, Liv, and I are all from Melbourne in Australia. So Melbourne located at the south end of Australia. And in case you're interested, it's currently 8 p.m. and it's also the middle of winter. And while Melbourne is a very beautiful city, it's also a city that's currently under stage 4 COVID restrictions, meaning that most of us haven't been to university for over 5 months. And at the moment, we're only allowed out of the house for an hour per day. So we've been under some sort of restrictions since March this year, and it feels very welcome to be able to connect with physiologists all over the world tonight. So to get into our topic, let's start with a couple of polling questions. So you will see 3 questions, and you will be asked to answer them in the next 30 seconds. And these questions are really for up to set up the scene, but also to gauge the audience and understand what level of understanding people have of muscle protein synthesis, which is the main physiological process we're going to discuss today. Okay, so you probably can see the results on your screen as I can. So this is really good because people know a bit about muscle protein synthesis, but not too much. And this is really important for us because now we know how to pitch our presentation, the level of our presentation, and no one's discovered AKT and mTOR, which is pretty reassuring. The second question was really to get us into the topic. How much sleep did you get last night? So it's quite reassuring to see that 62% of people actually got more than 7 hours of sleep, but it also means that 38% of people didn't get their 7 hours of sleep. And for adults, getting between 7 and 8 hours of sleep is the amount of sleep that's recommended to remain in good health. And this is actually very representative of what is seen at the world level. So here we saw 38% were more or less sleep deprived. And at the world level, or at least in Australia, it is estimated that 40% of people do not regularly get this 7 to 8 hours of sleep. And it's reassuring to see that most people did exercise in the last 24 hours, because one of the questions we're going to answer today is that if or whether sleep has a detrimental effect on our muscle, can actually exercise offset or reverse some of these effects. All right, so I think that we can really get started now. And let's talk about the relationship between poor sleep and skeletal muscle health and how people started to get interested in that topic. So I guess people started to investigate this relationship between poor sleep, which is defined as either poor quality or poor quantity sleep, and skeletal muscle health, as they discovered that sleep-deprived populations, and in saying sleep-deprived population, I'm sure you can think about a few of them, such as, for example, shift workers, but also young parents or the elderly are the typical populations that are chronically sleep-deprived. These populations are at a greater risk of developing metabolic impairments over time. And in saying metabolic impairments, I'm thinking diabetes, I'm thinking metabolic syndrome or sarcopenia. And in fact, these diseases are as much as 15 to 30% more prevalent in sleep-deprived population when compared to non-sleep-deprived population. And when talking about metabolism, we need to talk about skeletal muscle, because skeletal muscle is really a key player in whole body metabolism. So it's the largest organ of the body, and we all know that it plays a major role not in maintaining our posture and allowing movement, but it's also extremely important as a tissue that stores our nutrients. And most importantly, it's the major site, or one of the major sites of substrate metabolism in the body. So in summary, skeletal muscle is a fundamental component of our metabolic health. And the important thing to understand is that skeletal muscle is structurally made up of up to 80% of proteins. And therefore, muscle protein turnover, which is defined as the sum of the amount of protein that are being built up in the muscle and the amount of protein that are being degraded in the muscle, must be maintained in order to maintain skeletal muscle mass. That is, in order to avoid that the muscle shrinks or atrophied, and therefore to maintain muscle function and metabolic activity as well. And it is believed that poor sleep, through the dysregulation of the body biological clocks, may have the potential to impair this very important factor, that is muscle protein turnover. So where does this hypothesis come from? So firstly, it comes from observations that were made from population-based studies. And in these population-based studies, people have shown that poor sleep is positively associated with increases in the prevalence of muscle atrophy and sarcopenia. In other words, people who sleep less also tend to have smaller muscles and accelerated skeletal muscle, age-related skeletal muscle wasting, otherwise known as sarcopenia. And secondly, this hypothesis is also supported by rodent studies, which have shown that extended periods of sleep deprivation in rodents increase the markers of muscle protein degradation, also known as catabolism, and decrease the markers of protein synthesis, also known as anabolism, as well as directly decrease the muscle mass of these rodents. But at this point in time, nothing, or up until Nick's paper, nothing had been done in humans. And a few years ago, our group came up with a model to try and explain these relationships. And we started from the fact that muscle protein turnover, as you maybe know, can be regulated by 3 primary factors. So the first factor is muscle contraction, meaning that as you exercise, your muscle contracts and muscle protein are being synthesized. The second factor is dietary protein availability. As you eat, especially high quality protein, the same thing happens. The muscle protein synthesis pathway are being activated and your muscle can grow. And finally, the third factor is the hormonal environment, with some hormones such as testosterone promoting muscle protein synthesis, and some hormones such as cortisol promoting muscle protein degradation. And we realized that in situation of poor sleep, all these factors could potentially be disrupted in many ways. For example, because poor sleep alters not only light exposure, but also wake and sleep patterns and feeding patterns, all these factors have the potential to somehow disturb muscle contraction, protein availability, and the hormonal environment, and therefore the balance between muscle protein synthesis and muscle protein degradation, which is so important to maintain muscle health over time. So this leads me to introducing Nick's study. The first question he aimed to answer with his study was, does sleep restriction impair myofibrillar muscle protein synthesis in young healthy men? And since we said that muscle contraction is actually a very potent factor that has the potential to activate muscle protein synthesis, the second question that he aimed to answer was, can a protein synthesis stimulating batch or series of exercise, would it be able to offset or reverse this detrimental effect of sleep on muscle protein synthesis? And now it's off to Nick to explain more about his study design and results.
Hi, Sev. Thanks for the introduction. And so with that background Sev's given now, I'm going to talk a little more specifically about the study that we published earlier this year. But before I do that, I just want to say a quick thanks to all the other authors that were involved in the study as well, because they put in a lot of hard work over a number of years with this study too. So as Sev has alluded to, the rationale behind this was that we knew that sleep loss had been associated with changes in muscle mass before. We also know that muscle mass is regulated by changes in protein synthesis, and therefore we're interested in whether sleep loss could lead to changes in muscle protein synthesis too. And as Sev said, we know that exercise is a really potent stimulus for increasing muscle protein synthesis as well. And therefore we wanted to investigate if exercise would be useful in the context of sleep loss too. So therefore the aim of the study was to investigate the effect of sleep restriction with or without high intensity interval exercise on myofibrillar protein synthesis. Go to the next slide. So to investigate these aims, we conducted an 8-night sleep study at the Victoria University Sleep Facility. As you can see in the photos there, it's a fully contained unit. Participants had their own bedrooms. There was a communal kitchen, communal lounge room and bathroom, and it was next to the exercise physiology labs at VU there. So we recruited 24 healthy young males from around the university and matched them into one of 3 groups. We matched them on age, BMI, VO2 max, and also their habitual sleep patterns as well. Let me go to the next one. If you go to the next slide, Seb. Yeah. So,
sorry, there's a little bit of delay.
That's okay. This slide's going to show an overview of the study protocol that we used throughout the study. And this was based on a number of previous sleep studies which had used a similar design to investigate the effect of a lack of sleep on things like glucose metabolism. So we mirrored that somewhat. So we allocated our participants into one of 3 groups, either a normal sleep group, a sleep restriction group, and a sleep restriction and exercise group. And essentially all of these groups started the study in exactly the same way. They had an 8-hour sleep opportunity for the first 2 nights of the study. You want to go, Sev? However, after that, the normal sleep group, while they continued to have an 8-hour sleep opportunity for the next 5 nights, both of the sleep restriction groups only had a 4-hour sleep opportunity each night. And the only difference between the 2 sleep restriction groups was that the sleep restriction and exercise group also performed 3 sessions of high intensity interval exercise on the middle 3 days of the intervention. And so this exercise protocol was done on a cycle ergometer and it was quite intense. It involved 10 intervals of 60 seconds in duration, which was done at 90% of their peak power output as based on a pre-study VO2 max that we did. So in terms of the assessments there, what we did was we took muscle biopsies after the initial 2 nights of the study, and then again after the final night of the sleep intervention. And this was to help us to assess muscle protein synthesis throughout the study. And we did this using the deuterium oxide tracer technique, which I'll explain a little bit more about shortly. We also took a biopsy about a week before the study as well. And this was also to help with our calculations for determining muscle protein synthesis. So I'm just going to give you a little bit more detail now regarding exactly how we do these measurements and how we measure muscle protein synthesis as well. And I guess for those of you not from a muscle physiology background, I just thought I might show you what a muscle biopsy actually involves as well. So if you want to play the video, Sev. So this is a video of the biopsy for one of the participants in my study. And it's the tissue that they're obtaining there during a muscle biopsy that we use to do our analysis with. So once we've taken these samples, if you want to go to the next bit, Sev. As I mentioned, our main outcome measure for this was assessing myofibrillar protein synthesis or muscle protein synthesis. And the reason we were so interested in this is it's because it's been shown that it's the rates of muscle protein synthesis which are known to underpin changes in muscle mass. And that is opposed to instead of, I guess, looking at rates of protein degradation. And so traditionally, muscle protein synthesis has been measured using amino acid tracers, which basically requires or allows you to measure the incorporation of these amino acid tracers into the muscle as a measure of muscle protein synthesis. However, this sort of procedure has generally required that participants remain in the laboratory for the whole time that you're measuring protein synthesis, and therefore can only be measured over short durations. However, because of those reasons, a number of physiology-based studies recently have started using the deuterium oxide tracer technique to measure protein synthesis. So deuterium oxide, or D2O, is basically deuterated water, has an extra neutron in there, which allows you to track its incorporation into new protein that's synthesized. And so essentially what this does is allows you to measure muscle protein synthesis in free living conditions and gives you a really good integrated measure of protein synthesis in a number of different situations, such as the fasted and fed state, after exercise, or while you're asleep, for example. The other advantage of using this method is it's really quite a simple method to implement into your study, and our study in particular. So essentially what happened was the participants would arrive for the first night of the study. They'd be given a drink of D2O, which is just basically a glass of water. We give that some time to equilibrate within the body and then take our first muscle biopsy. So after that, the participants go about the intervention as we showed you before. We're sleeping either 8 hours or 4 hours a night. And then we collect another muscle biopsy on the final night. And then from this, we can then use mass spectrometry to assess the amount of the deuterated water which has been incorporated into the muscle and therefore protein synthesis. And really all else you need to do during the intervention is collect saliva samples to monitor the amount of deuterium oxide. So aside from that measure of protein synthesis, it's also quite common to look at these using molecular markers which are known to regulate protein synthesis and also degradation as well, get an idea of what's happening with muscle protein breakdown. And so we looked at things like AKT, mTOR, P70S6K, protein synthesis pathway, and also markers of protein degradation, just using traditional markers like traditional techniques like real-time PCR and Western blot. Thanks, Sev. Thanks. So I'll start to show you a few results. And I guess the first thing we need to show you is that those in the sleep restriction group actually slept less than those in the normal sleep group. As you can see in the table here, during the baseline period, the initial 2 nights, all groups slept for a really similar amount each night. But then you'll see during the intervention, both sleep restriction groups slept for significantly less than the normal sleep group. I've also got on there sleep onset latency. And sleep onset latency is actually really just a measure of how long it takes the participant to fall asleep once they go to bed. And as you can see there, those in the sleep restriction group fell asleep pretty quickly. And so we measured sleep just using wristwatch actigraphy, but we also confirmed it with some polysomnography as well. Thanks. So this is the main outcome of the study really. And this graph shows myofibrillar protein synthesis as we measured with the D2O tracer technique across the entire intervention. And as you can see there, the sleep restriction group had a significantly lower rate of myofibrillar protein synthesis compared to the normal sleep group. But you also see that those in the sleep restriction and exercise group actually maintain their level of myofibrillar protein synthesis to a level comparable to the normal sleep group. And so we think that these results are actually really important findings. It's because of these changes to things like muscle protein synthesis, when they happen continually, is what are actually going to lead to changes in muscle mass over time. Next slide. Thanks. So aside from, because of the results we'd seen, I guess, in the protein synthesis, we then assessed the protein content of the molecular signalling pathways that we know that regulate the protein synthesis process. However, despite the changes in protein synthesis that we saw in the last graph, we actually didn't see changes in the molecular signalling pathways that corresponded to these changes in protein synthesis. And likewise, when we measured molecular markers of protein degradation, we also didn't see any changes from pre to post intervention. So these graphs here just represent pre to post changes from the Western blots that we did for the main regulators of the protein synthetic pathway. So as you can see, we saw no change in either total protein or phosphorylated protein in AKT or mTOR or P70S6K either. Now we go to the next slide. So we also wanted to have a look at the gene expression of some of these molecular pathways as well, and to see if they show any change. However, like the protein content that we just showed you, we also actually saw no changes in the gene expression of genes known to be involved in the maintenance of protein synthesis and also in the degradation pathways. Again, these graphs here show pre to post changes in gene expression. For example, myostatin mRNA, which is known to be an inhibitor of the protein synthesis pathway, we saw no change in that either. Go one more there, Sev. So in summary, what we showed was for the first time here that rates of muscle protein synthesis were lower following a period of sleep restriction, which consisted of 4 hours sleep a night for 5 nights in a row. But there's a number of questions I think we can still ask regarding this and which we don't have answers for yet. We need to explore these a little bit further. For example, if these changes in protein synthesis were to persist, or would they persist over extended durations with continuing sleep loss? And would it also lead to a reduction in muscle mass? We're also interested to know how do these results actually translate to the sort of sleep loss people commonly experience in the community. We know that the Australian population, up to 20% of that population sleep less than 6 hours a night. So how do these results actually translate for them? The next thing we showed was that performing high intensity interval exercise during the sleep restriction period prevented the decrease in protein synthesis that we saw. So I guess an obvious question here would be, what do other exercise modalities do? So we used endurance exercise here, but what would things like resistance training do? And another prominent question, I think, is we didn't actually show whether the sleep restriction protocol actually inhibited the increase in protein synthesis that exercise has been shown to induce previously. And so if we'd had a normal sleep and an exercise group in this study, we potentially could have investigated if there's any inhibition caused by the sleep restriction protocol. So the last thing is that despite the changes in protein synthesis that we recorded, we didn't see any changes in the molecular signaling pathways that regulate protein synthesis or that regulate protein degradation. But I think this actually isn't too surprising, I think. The measures that we took and looked at using PCR and Western blot, they're actually just static snapshots of the muscle at the exact time that you take that muscle biopsy. And so they don't necessarily reflect the changes in these pathways which occurred throughout the 5 days of the intervention. And for example, if we'd taken a muscle biopsy at different times throughout the study, for example, after the exercise, we may well have seen changes with these molecular pathways, which might explain the integrated protein synthesis response that we saw overall. So in summary, I guess these results provide novel insights into the previously described changes in muscle mass that have been attributed to inadequate sleep. And we've also showed that high intensity interval exercise can potentially mitigate or counteract these effects in this context too. So I'm going to leave it there and I'm going to pass you on to Liv, who I think is going to discuss this work a little bit more and some of the implications with it as well. Thanks.
Thanks, Nick. So as Sev mentioned previously, I wrote a Journal Club article titled "No Time to Sleep, Start Exercising" that aimed to unpack the findings from Nick's paper, as well as discuss the next steps for research in this area as well. Since my article, another journal article has been written that discusses, Journal Club article has been written that discusses the different perspectives on these results. There is some crossover between both our articles. However, Estefanos and colleagues do discuss regulators of muscle mass at a cellular level to a greater extent. So both articles are certainly worth having a look at when you get the chance. If you can jump to the next slide, please, Dev. So Nick just touched on the pathways that regulate skeletal muscle protein synthesis, and I did just want to recap how we can look at the possible mechanisms of the findings from Nick's work. So firstly, as Nick alluded to, their findings represent a snapshot in time, and since there was no change found in the molecular markers in the study, the next step would be to observe these markers, as Nick said, at a different point in time. The second step would be to understand what additional markers of muscle protein synthesis we can observe. So measuring muscle protein degradation directly is possible given that we know that from the animal models that Sev mentioned earlier, they have shown that sleep deprivation can increase rates of muscle protein breakdown. However, muscle protein degradation is particularly difficult to measure and is likely a contributing reason as to why Nick and his colleagues didn't measure this. The next option could be to look at the role of hormones. So if you have a look at the right of this diagram, we know that we can standardize muscle contraction by controlling the exercise and daily physical activity that participants perform, which Nick and his colleagues did. We can also standardize amino acid availability by ensuring a standardized feeding protocol, which leaves our hormonal response, which we obviously don't have the same control over. So as depicted by this diagram, we know that the key anabolic and catabolic hormones such as testosterone, IGF-1, growth hormone, and cortisol play a really key role in regulating muscle protein synthesis, and that these hormones are also regulated by our sleep and our circadian rhythms. So, for example, research has shown that testosterone decreases and cortisol levels increase with sleep deprivation. Therefore, this could in turn blunt the muscle protein synthesis response. So certainly each of these factors are worth investigating in future research, which I guess takes me to the next slide, please, Sev. And in terms of the implications for future research and how the findings from Nick's work are clinically relevant for sleep-restricted populations. The notion that we can rescue muscle protein synthesis by using an exercise intervention is really important, and the way we deliver and prescribe that exercise is equally as important so that we can optimize the translation of these findings into practice. So Nick mentioned that him and his colleagues used a high-intensity interval training model that, in addition to increasing muscle protein synthesis, also has cardiometabolic advantages. They also used a really time-efficient model, which is important when a lack of time is one of the biggest barriers that we see for populations trying to perform exercise. However, it's also worth considering that if increasing muscle protein synthesis in order to maintain or improve muscle mass is our main goal for sleep-restricted populations, then resistance training might be a good alternative for sleep-restricted populations. Metabolic response to exercise is mode-specific, and resistance training is known to be the gold standard mode of exercise to increase muscle protein synthesis. This is in part due to the fact that myofibrillar protein synthesis is particularly sensitive to mechanical loading. So the load that is placed on the muscle during resistance training triggers muscle hypertrophy via accumulated bursts of muscle protein synthesis. In contrast, high-intensity interval training or aerobic exercise is typically performed under a lower load and has a different pattern of neurological activation, muscle fiber recruitment, and energy expenditure pathways, etc. As a result, understanding the role that resistance training might play in this type of sleep restriction protocol will be important for practitioners looking to apply these findings in the real world. Can you jump to the next slide, please, Sev? So once we're able to determine how to deliver a best practice intervention, it's also important to consider the population groups that we can apply these findings to. So Nick's work was performed in untrained participants, which is certainly appropriate, but if as a result of these findings, we are then prescribing regular exercise to sleep-deprived populations, after several months, those people will then become what we consider trained participants or trained athletes. And therefore, given there is evidence to suggest that the muscle protein synthesis response to exercise could diminish as we adapt to training, it's worth understanding if either a high-intensity interval training model or a resistance training protocol can, again, rescue that muscle protein synthesis to the same extent in trained participants. My final comment is that it remains really important that we are also inclusive of female participants in sleep and exercise research, and for a couple of reasons. The first is that we know females report more sleep problems and need and have a need for longer sleep duration than males. And given that females are equally represented in populations who are likely to be exposed to inadequate sleep, if we think of our shift workers, new parents, this makes them an important group to investigate this type of protocol in. The second is that females are not just small males, and their menstrual cycle plays an important and pretty unique role in regulating their sleep, their circadian rhythms, and the skeletal muscle hypertrophy response as well. So while there's certainly more research that's needed in this space, there is evidence that REM sleep is sensitive to menstrual cycle phase, and additionally estrogen, which is a key hormone that regulates and fluctuates across the menstrual cycle, also regulates the AKT-mTOR pathway and could therefore play a key role in females' capacity for muscle protein synthesis across the menstrual cycle. Finally, females might have a blunted myofibrillar protein synthesis response to high intensity interval training compared to males. Therefore, it would certainly be interesting to understand if high intensity interval training can rescue muscle protein synthesis to the same extent in females with the protocol that Nick and his colleagues used. This might also be another argument for the use of resistance training, given that males and females have a similar capacity for strength gains based on the current resistance training literature. Overall, what I hope you've interpreted from these final slides from our presentation is that the findings from Nick's work are really important for populations who experience inadequate sleep, and that the next steps we take toward optimizing the translation of these findings into practice is really exciting. That's it for me, Sev. Thanks.
Yeah, so that's it for us. Thank you very much, everyone, for your attention. And I guess we'll open the room for questions now. I can see that a couple of questions have popped in the chat already. And before we get onto the questions, just remind you about our virtual networking event, which will take place after the question. And I believe that Imran will post the link in the chat box when appropriate. So let's have a look at these questions now. Okay, so we have let's start. We have a couple of questions in the chat, and then I'll get to the Q&A. So someone actually made a comment saying, I thought the skin was the largest organ in terms of area. So I think we're talking in terms of weight here, really. That's my only answer to that. Does anyone want to add anything else? No. Okay. So let's say skeletal muscle is the largest organ of the body when talking about its volume or weight. A question that I believe has been answered when Nick talked about his limitation. Does protein synthesis in people increase in people who don't have sleep restriction? And I believe, Nick, you have touched on that.
Yes, sorry. Yeah, absolutely. And maybe I didn't make it as clear at the start as I could have, but there's a number of studies that show exercise protocols such as that we used in our study induce an increase in protein synthesis. Yep. It's a fairly well documented response to a session of either high intensity training or resistance training, and usually occurs for sort of 24 to 48 hours after the exercise bout. Yeah.
Thanks, Nick. I can see that someone's raised their hand, so I'll give them priority, and it's actually Brad. Hi, Brad.
Hi, Sev. Great presentation, Nick and Liv. I had a question, Nick, for you about motivation during the high intensity interval training. We know that inadequate sleep drops motivation. How hard did you have to coach the participants on maintaining high intensity intervals?
Yeah, that's good. That's a fair question. And it definitely varied between participants. Some of them thought, yeah, great, this is a great way to get through the day. We'll do some exercise and hopefully it'll perk me up a little bit. Others were sort of more of a little bit of dread. Oh, do I have to do some exercise? So I think it's a fairly good point that this is always going to be a personal preference in these types of situations. Yeah, I think that's it as well.
Thanks, Nick.
Thanks,
Maybe
Nick. Thanks.
Liv, did you have a comment given your bent in your focus on exercise?
I guess from the research and the studies that we've been conducting, we've tried to control motivation as much as we can from a researcher perspective by only giving them certain cues and not trying to encourage them too much, so that the motivation that we're providing is somewhat standardized. So to try and control for that as well.
Great, thank you.
Thank you. So now on to the Q&A. So we'll just take them chronologically. So a first person who remained anonymous asked whether does treating the cause of poor sleep normalize muscle protein synthesis? That's a very good question. Nick, do you have an answer to that?
So I assume you mean after, you know, a night of recovery sleep or something like that, does the rate of protein synthesis return to normal? And I guess the answer is that I don't think anyone knows. You'd speculate that whether it does after one night of recovery sleep or a couple of nights, it obviously goes back to where it was at one point. And I guess that also plays into the fact we don't know exactly, you know, if you continue to sleep only 4 or 5 hours a night, will the protein synthesis response remain lowered? And I don't think, we just don't know that at this point.
The next question is from Sarah Alexander, and she's asking whether we have a hypothesis about whether sleep restriction would blunt the muscle protein synthesis response to exercise. So Sarah, this is the whole point actually, and Liv has touched on that on her mechanism slides, because at the moment, Nick's results are obviously super interesting, but it's a bit hard to tease out the underlying mechanism. And I think Liv's done a very good job at explaining that basically, if you look at the 3 factors the 3 primary drivers of muscle protein synthesis, we've got exercise that can be normalized to an extent, or muscle contraction. We've got dietary protein intake that can be normalized. And the one that cannot be normalized in an experimental setting is the hormonal environment. So my hypothesis is that the hormonal environment has something to do with that blunted response. Now, we don't know if it's the only mechanism at play. We don't know to which extent. And as you might be aware of, it's actually tricky to test experimentally. The next question was from Simon, and Simon is telling us that there is plenty of evidence from behavioral ecology that non-human animals in the wild optimize how they divide the time between different activities. Do humans do the same? Liv, do you want to give a try at this one?
I don't really know the answer to this question. I mean, I suppose at this, we don't, as humans, we're not really used to splitting up our sleep unless you are napping, I suppose, in terms of our sleep activities. And we do know that napping and sort of supplementing your sleep, if you are restricted, can make a difference in terms of your fatigue levels, but I'm not quite sure what that role would play in terms of muscle protein synthesis. I don't know if that answers the question.
Yeah, I'm not sure either, but it's a good question that is certainly worth investigating in the future. So a question that I believe has been answered. So Jay asked whether the decrease in protein synthesis induced by sleep deprivation can be reversed by exercise. And I think, Nick, we can say that everything in your paper points out towards a yes here. Do you agree?
I think so. I think that's, that's probably is what we've shown, as opposed to knowing whether or not the sleep restriction is blunting the MPS response itself, in terms of if it's inhibiting an increase in muscle protein synthesis that we'd usually see to exercise. But it does look like exercise is able to at least maintain levels of protein synthesis similar to a normal sleep amount.
Thanks, Nick. Then we have a question from Brian, who is from the University of Sao Paulo. He has a question for Nick, but I'm actually going to answer this one. What do you think the effect of endurance exercise might be for muscle protein synthesis during sleep restriction? So, Brian, to me, and we've had this conversation with Nick a couple of days ago, and I was wondering why they hadn't chosen a resistance exercise intervention in order to maximize the amount of muscle protein that would be synthesized during the sleep restriction period with exercise. And the answer was simply that this study was part of a larger study with investigating other outcomes. But anyway, Liv and I a while ago wrote a review. What did we write? We did write something which led us to investigate what was the effect of endurance exercise on muscle protein synthesis. And we found that why not as powerful or potent in activating muscle protein synthesis, endurance exercise is muscle protein synthesis stimulating still. And I don't know, Liv, do you remember to which extent or to which percentage this type of exercise was able to activate the muscle protein synthesis pathways?
I can't remember off the top of my head, but I can have a quick flick through that paper and try and just put it in the chat if I find it. Sorry.
Yeah. So basically, short answer is endurance exercise still activates muscle protein synthesis. Remember that in this study, for reasons that Nick has explained, he hasn't looked into the effect. He hasn't looked at the effects of sleep deprivation on muscle protein degradation, and that's where the difference with endurance exercise is, because we know that endurance exercise, yes, does increase muscle protein synthesis, but it does also increase muscle protein degradation, and that's why you do not hypertrophy when only performing endurance exercise, and that's where the difference probably lies. But in that case, yeah, it's still able to activate the muscle protein synthesis pathway. Then we have Rowan who is from the University of Portsmouth, and he is asking, that's one for you, Nick, why did you choose the 4-hour sleep opportunity, considering that less than 7 hours would already be considered sleep restriction or partial sleep deprivation?
Yep, that's a fair point. So for a number of reasons, I guess, to start with, 4 hours sleep has been a fairly constant amount of sleep in a number of previous sleep studies that have looked at different effects of sleep loss on metabolic health. So the original studies that we looked at and read investigating changes in insulin sensitivity and glucose tolerance used 4 hours sleep a night. And so we just were trying to mirror that. We also wanted, I mean, in a lot of ways, this is really just a proof of concept type study. We wanted quite a severe sleep restriction. And a number of the previous studies that had been done in animal models that actually use really extreme sleep deprivation, so upwards of 96 hours of sleep deprivation to see the changes that they saw. So we still wanted to keep quite a severe sleep restriction. But the further I got through this study, the more I realised, though, that there are actually a number of populations out there that are sleeping 4 hours a night. You know, it might not be every night, but people are definitely going through periods where they're not sleeping very much. So it seems drastic, but it's actually not that far from the truth in some instances.
Thanks, Nick. So I don't think we'll get to all of the questions, so we'll take another 3 in the order they pretty much appeared. And then Imran suggested that he could email us the rest of the question, and maybe we can try and provide an answer via email over the next few days. So we had a couple of questions that appeared early on in the chat, and I'd like to bring them up because they're actually quite interesting questions. So Ollie from King's College London is thanking us for sharing this great study. And he's asking you, Nick, if you did also measure mitochondrial muscle protein synthesis rates given the HIIT nature of the exercise. And if you haven't, what would you expect to see with sleep restriction, with or without HIIT?
Yeah, it's a really interesting question. And yeah, so part of our study was actually also aimed at looking at some of these aspects. So we're interested in things like glucose tolerance and mitochondrial function as well. And so as part of this study, or as a part of the study as a whole, we attempted to look at mitochondrial protein synthesis. We actually ended up measuring a proxy of mitochondrial protein synthesis and measured sarcoplasmic protein synthesis, just due to restrictions with the amount of muscle that we actually got with the muscle biopsies at times. And so we're still trying to get that data out at the moment, but there's a preprint available. And we did see a reduction in sarcoplasmic protein synthesis with sleep restriction, which was mitigated with HIIT. Yep.
Thank you. And there was a follow-up question from Ollie, who was asking whether the protein feeding patterns, that is the dose and timing distribution during the day, change with sleep restriction. I may have missed this in the presentation, perhaps you controlled. Can you comment?
Yeah, no, and they're both good points as well. We standardised the diets for all participants. I think we had 1.5 grams per kilo of protein, I think off the top of my head. And what each participant had was we standardised that we had 6 eating opportunities each day, breakfast, lunch and dinner, and then 3 snack opportunities, all at set times throughout the study as well. So we tried to control that as much as possible too.
Thanks, Nick. There's one question for you, Liv, here. Sienna is asking whether there's such thing as improved sleep quality that is independent on sleep duration, and whether does exercise improve quality of sleep, and could this contribute to increased muscle protein synthesis?
There is a systematic review available that does discuss the effects of exercise on sleep. And I probably know more about the effect of resistance exercise on sleep quality and quantity, but in that scenario, it certainly does improve sleep quality in particular. Was there a second part to that question? I've lost it. Oh, you've...
whether sleep, yeah, now I've lost it too. Whether sleep can actually improve, whether exercise can actually improve sleep quality.
Yes, so we, that systematic review that I'm referring to did find that exercise can improve sleep quality. So yes, is the answer.
Yeah. So as I say, we won't have time to go through all the questions, so I'm going to be really biased here, and I'm going to answer one that I want to answer. How much of an influence, and that's Emily Rogers asking, how much of an influence do you think cortisol might have on the changes in muscle protein synthesis? And do you think it would be worth measuring in future studies? So Emily, you're touching the exact point that we mentioned earlier about the mechanism. So cortisol has more of a catabolic effect, not an anabolic effect. So basically, cortisol is expected to increase muscle protein degradation rather than decrease muscle protein synthesis. Also in animal models, cortisol can decrease muscle protein synthesis, but it hasn't been shown in humans. In humans, it is believed that it has more of an effect on muscle protein degradation. And we come back to this idea that muscle protein turnover is the sum of muscle protein synthesis or degradation. So on the long term, it's speaking of the long-term implications for muscle mass and muscle metabolism as a whole, yeah, it would be really important to tease out whether cortisol actually plays a role in muscle catabolism. And very last question from Natalie, which will be of interest for you, Liv. Question related to females blunting myofibrillar protein synthesis response to HIIT. Would resistance work better for female?
I think we don't know the answer to that yet, and hopefully some of my work will with Sev will be able to answer that question. Yeah, we don't know the answer, but given that females and males respond similarly in terms of their adaptation to resistance training, hopefully resistance training might work better, I guess, than high intensity interval training in the sense of stimulating muscle protein synthesis.
So as Liv said, keep an eye open for her amazing publications that will come up one day when we are out of lockdown and we can actually resume our studies. But that's exactly what she's investigating. She's investigating the potential effect of sleep restriction in females. and whether protein stimulating types of exercise, such as resistance exercise, are able to blunt or reverse any potential impairments. So it's nearly 9 o'clock here. So I'd like to thank everyone for your participation. So a few people, actually quite a few people, asked questions that we didn't have time to answer tonight, but we'll take a copy of these questions and we'll get back to you via email in the next few days. Now, I'd just like to remind you that we are now holding a networking event, and if you go back to the chat, I will repost the link that Imran has posted at the very start. So please join us. It's a short 25 minutes maximum networking session, and we'd really like to see you there. And again, thank you very much for attending tonight. Thank you very much for your great questions. And if you want to get in touch about anything, PhD, postdoc opportunities, research ideas, do not hesitate to email us directly. Thank you very much, and have a good day for those who are in Europe, and have a good night for those who are in Australia. And I hope to see you in the other Zoom room. Thank you.